This talk is inspired by a fundamental principle of Baha'i faith. It represents my perspective, my reflections on the Baha'i principle of the harmony of science and religion, a theme relevant to our times. At this beginning, I wish to offer few introductory words on the Baha'i faith, the most recent of world religions. The principles and teachings of Baha'i faith inspire and guide this global age. Baha'i faith is widely spread across the entire planet, from pole to pole and in all corners of the world, including within its fold members from diverse races, religions, nationalities, and ethnicities. It is a veritable microcosm of humanity dedicated to the principle of the oneness of humankind. Thus, it is well suited for bringing about the unification and solidarity of the races, ethnicities, religions, and nations across this planet. This talk is centered on the Baha'i principle of the harmony between science and religion. Among other fundamental principles of Baha'i faith are independent investigation of truth, elimination of all prejudices, equality of women and men, the oneness of the human race, the recognition of the validity of the major religions of the world, and the dynamic and progressive interrelationship among them. For the first time in the history of religions, the Baha'i faith, a world religion, pronounces unequivocally the principle of the harmony of science and religion. This fundamental principle is at the center, at the core of the very heart of this presentation. The harmony of science and religion is an intriguing, a fascinating principle, which for long has been sidestepped and evaded. Now is the time to reflect on this principle. Among the world's great religions, the Baha'i faith unequivocally promotes and champions the harmony of science and religion by designating it as one of its fundamental principles. This presentation draws attention to the ethical challenge posed by biotechnology, in particular by the biotechnology of genetic engineering, challenge to the principle of the harmony of science and religion. May this presentation generate such interest as to inspire the viewer to explore, investigate, and reflect on the Baha'i faith and its teachings that are precisely in tune with this global age. First, I wish to reflect on the Baha'i principle of the harmony of science and religion, and next, to bring to the attention of the viewers the ethical challenge posed by biotechnology, namely the biotechnology of genetic engineering, to the Baha'i stance on the harmony of science and religion. This ethical challenge compels reflection. Reflection on the ethical challenge posed by biotechnology, notably by the biotechnology of genetic engineering. Such reflections lead to a fundamental question: are science and religion in opposition to one another, or are they in harmony with one another? Science and religion each has been a significant force in human history, essential to the development of society, civilizations, and cultures. Yet often, science and religion are viewed in opposition to one another. Science and religion are as two mighty forces, as two waves surging toward the development and advancement of civilization. Thus, harmony of science and religion inspires reflection. Reflection on the harmony of science and religion. Science and religion are as two waves, seemingly divergent waves, that originate from the same source. They arise from the same source as two mighty waves. These two waves, religion and science, cannot be separated. They are intertwined with each other. They are as two waves, two mighty forces in the development and progress of civilizations. Two sweeping, divergent waves arise from the same source. The sweeping wave of science and religion originate from the same source. Religion and science are not separate from each other. Rather, they are intertwined. Indeed, they may be viewed as mighty waves that move onward as convergent waves. They progress toward the advancement of civilization. Should these sweeping waves converge in harmony, they can generate a tremendous force, lending a powerful impetus to the advance of civilization. Historically, science and religion have been viewed in opposition to one another. I wish to emphasize that the fundamental principle of Baha'i Faith, the harmony of science and religion, is emphasized and affirmed by the following few selections from the Baha'i sacred writings. Religion and science are intertwined with each other and cannot be separated. These are two wings with which humanity must fly. Religion and science must be reasonable. It must agree with science perfectly, so that science shall sanction religion and religion sanction science. If we say religion is opposed to science, we lack knowledge of either true science or true religion, for both are founded upon the same premises and conclusions of reason, and both must bear its test. Enlightened and inspired by these writings, we can pose the key question: are science and religion dissonant or in harmony? Science and religion, dissonant or in harmony, is a question that over centuries has been the cause of much contention, a contention that has continued to the present, a time when great advances in science, technology, and innovations have been achieved. A related question is, are science and religion at odds with one another, or are they complementary and supportive of one another? Both science and religion are centered on what is believed to be the truth, in one respect relating to the physical phenomena, and in other respect relating to spiritual truths. Thus we ask, are science and religion in competition, or in cooperation, or in synergy? Are they complementary and supportive of one another? We can probe the question of harmony and science and religion through several approaches. Traditionally, for long, it was assumed that science and religion are at opposing poles and that never shall the twain meet. Science relates to the operation of those universal laws of nature. For those who believe in a religion, religion relates to spiritual laws and principles. One of the fundamental reasons that religion and science have been viewed in opposition to one another is due to lack of understanding or misinterpretation of metaphors, analogies, and similes used in religious texts. These have been taken literally, though they hold profound insights and have layers of meaning, whose depths may be fathomed through thoughtful reflection. Lack of understanding of numerous metaphors in the Judeo-Christian, as well as other sacred texts, Zoroastrian, Buddhist, Hindu, and Muslim, are factors that have contributed to such misrepresentations. Historically, on one hand, contention between science and religion has deterred women and men of science from partaking of the wisdom and riches that religions can impart. On the other hand, it has hindered many women and men dedicated to a religion from enrichment that science can impart toward understanding the mysteries of life and enhancement of the quality of life. At one time, a talk on science and religion would have likely centered on a literal understanding of certain religious texts, such as the story of creation in 7 days, the Garden of Eden, the question of evil, and numerous other examples. Abdul Baha, one of the central figures of Baha'i Faith, sheds insights on these themes through his talks and writings. Some of these are to be found in a remarkable book known as Some Answered Questions. He sheds insights by explaining the inner meaning of these metaphors. Yet there are those who insist on the inerrancy of a literal understanding of the sacred text, such as the story of creation, exclusivity of salvation, and others. In our times, the literal understanding of such metaphors no longer merit serious consideration. Interpretations of sacred texts go well beyond the literal. Now, in our times, in current times, we are beyond the literal understanding of such metaphors and analogies. There are novel challenges to be met in our times. In particular, advancements in science and technology compel insightful reflections on the Baha'i principle of the harmony of science and religion. The Baha'i principle of the harmony of science and religion. During our time, special challenges are posed to this principle, in particular by the ethical challenge posed by biotechnology, notably by the biotechnology of genetic engineering. This prompts an in-depth reflection on the biotechnology of genetic engineering. That is regarding the ethical and spiritual standpoint in bringing together material and spiritual civilization. I quote from Baha'i sacred writings: although material civilization is one of the means for the progress of the world of mankind, yet until it becomes combined with divine civilization, the desired result, which is the felicity of mankind, will not be achieved. In our times, momentous advancements have been achieved in biotechnology. This prompts us to ponder whether biotechnologies hold prospects for human well-being. Their benefits are significant, among them identification of the genetic base of disease, eradication of certain genetic diseases, development of therapies, prolongation of human life, and much more. Or whether biotechnologies foreshadow unfavorable outcomes toward human detriment. Few among their unfavorable, even ominous implications are human cloning, eugenics, and generation of biohazards. As the gene is at the foundation of heredity, we are faced with multitude of issues and concerns. At this point, it is important to first briefly overview some of the historical background on human heredity. Early groundbreaking studies include the work of Charles Darwin with its implication on inherited traits, presented in 1856, the work of Gregor Mendel on P hybrids, presented in 1863, William Bateson foresaw that the science of heredity would lead to great discoveries and that its power would be applied to control the composition of a nation. Thus, Bateson introduced the concept of eugenics. Whether such control would ultimately be good or bad for that nation or for humanity at large was considered a separate question. Then Francis Galton coined the word eugenics in 1904. Eugenics may be defined as the science of improving a human population by controlled breeding to increase the occurrence of desirable, heritable characteristics. Eugenics has horrific implications. A few years later, in 1912, Alfred Plotz, a German physician, biologist, eugenicist, coined the term racial hygiene and promoted the horrific concept of racial cleansing in Germany. In fact, it needs to be said that the United States has also been tainted in respect to eugenics. Genetic cleansing was promoted between 1927 and 1972 through the sterilization of some women identified as imbeciles. Regarding this case known as Buck versus Bell, 1922, the US Supreme Court actually ruled that Virginia's law was constitutional and that Carrie Buck should be sterilized. Her sterilization was the first of approximately 8,300 performed under the law between 1927 and 1972. Eugenics has horrific implications. At this point, let us take a brief look at certain landmarks in the development of the science of heredity. Early on, Aristotle recognized that heredity is as a current of information moving across generations. Gregor Mendel, in 1866, introduced his work, resulted in what is called Mendelian genetics on pea plants. The discovery of the structure of DNA in 1953 was revolutionary. It led to molecular genetics. Molecular genetics has ever since dominated the field of genetics with its revolutionary effect, leading to the development of some remarkable biotechnologies. With the emergence of molecular genetics, we are faced with a challenge that on one hand are auspicious with immense possibilities, yet on the other hand, they also pose the dilemma. Thus, they pose both the challenge and the dilemma. The new discoveries in biotechnology were hailed with great enthusiasm by some passionately as the ultimate experiment, man-made evolution, the brave new world of genetic engineering. The brave new world refers to a dystopian novel written by the English author Aldous Huxley in 1931 and published in 1932. This book was published some 20 years before the structure of DNA was deciphered. Aldous Huxley foresaw that which was on the horizon could lead to a dystopian future with its horrific implications. Yet others foresaw the new findings auspicious, a boon to humankind. In time, it led to the discovery of the structure of DNA in 1953. That discovery was enthusiastically hailed as alchemy revived, improving nature, harnessing the gene, and remaking the world. Why such discord? Why such discordant views? Which of the two did the new discoveries present? A blessing, boon, Or was it dismal, gloom? Which is it? Could it be both? Gloom and also gloom, as it relates to the theme of this presentation, the harmony of science and religion, boon and also gloom? First, I plan to briefly overview the boon, that is the advantages, favorable outcomes of this technology, that is the benefits, life-saving benefits of some of the discoveries, advances at the forefront that are possible through biotechnology. Next, I want to reflect on the possible disadvantages, the unfavorable outcome of biotechnology, that is the harmful effects which can last over generations, extending into the future generations. Now, an overview of certain advances at the forefront in biotechnology. Some of these advances are a boon. They hold outcomes that are beneficial to human well-being. Some others are dismal, gloom. They portend detrimental outcomes. Now, an overview of advances at the forefront. The discovery of the structure of DNA in 1953. Several biotechnologies, among them recombinant DNA, gene cloning, stem cell research, and genetic engineering hold prospect for human well-being, yet can also have detrimental outcomes. Their significant benefits include identification of the genetic base of disease, development of therapies, prolongation of human life, and much more. However, among their unfavorable implications are eugenics, gene enhancement, human cloning, and the generation of biohazards. These advances, with their significant benefits as well as their unfavorable implications, are the outcomes of the discovery of the structure of DNA in 1953. The discovery of the structure of DNA was revolutionary. In 1962, Nobel Prize in Physiology and Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins, but left out a principal figure, Rosalind Franklin, who had died in 1958 at the age of 37. This is exciting, a fascinating story, yet it is egregious. It is fascinating because of how it happened. That aha moment, that eureka moment of sudden discovery. It is egregious because the process ultimately left out the major contributor, Dr. Rosalind Franklin, as one of the recipients of the Nobel Prize. Dr. Rosalind Franklin had died before the prize was awarded. The Nobel Prize is not awarded posthumously. I wish to relate the fascinating story of this monumental discovery, leading to the eureka moment, that sudden discovery. How so? Researchers in the field, James Watson and Francis Crick, were conducted by Maurice Wilkins, the director of the lab at King's College in London, and shown the crystallographic data studies of Dr. Rosalind Franklin. Let us now look at that key study, the fundamental data, the key image which divulged the structure of DNA. The discovery of the structure of DNA was based on the data, the research of Dr. Franklin, this crystallographic X-ray diffraction image of DNA. Viewing this image led to that eureka moment for Watson and Crick. Over the years, various models of DNA had been proposed as a single strand or a triple strand, and so on. However, it was the data, the crystallographic image of DNA of Rosalind Franklin, which at long last divulged the model of DNA. it became suddenly clear that DNA must be in form of two strands, the double helix. How so? This was based on the observation of this image, the crystallographic image of DNA, obtained through these studies. The distinctive X in this image divulged the pattern of a double helix. James Watson and Francis Crick became excited and giddy after viewing this image. They now had the view of the essential image of the molecular structure of DNA. They had that eureka moment. Immediately, they perceived the overall organization of the structure of DNA. Over time, eureka moments have sparked scientific discoveries. Some of us may have experienced a eureka moment when suddenly we become aware of a long search for reality, whether a scientific discovery or a spiritual discovery. Through model building, things fell into place. The double helix structure of DNA of two strands was at long last disclosed. So what did James Watson and Francis Crick do? Through model building using hammers and screws and metal plates, they unraveled the organization of the double-stranded model of DNA, the double helix. There is a photograph of that model building, which shows James Watson and Francis Crick in state of awe and euphoria. All that model building. James Watson and Francis Crick are credited with deciphering the structure of DNA. Then James Watson and Francis Crick went to the Eagle Pub in Cambridge, near Cavendish Laboratory, to celebrate. A sign has been put up by Eagle Pub in Cambridge, acknowledging and celebrating that historic event. This pub, to this day, bears the distinction of its association with the discovery of the structure of DNA. They used the wording for DNA double helix, the secret of life. That was where they first announced the discovery of DNA, carrier of genetic information. So that was how it happened. That was what happened on February 28, 1953. The double helix, the secret of life. Having related this story, in the interest of simplicity, I need not show in this setting the rather complex yet magnificent molecular image of the double-stranded DNA. Therefore, in the interest of avoiding undue complexities in this session, I will bypass showing and explaining the diagram of the molecular structure of DNA. The discovery of the structure of DNA, the secret of life, has had a great impact, a revolutionary impact. Once the molecular structure of DNA was deciphered, it led to the development of biotechnologies with their immense possibilities. Biotechnology holds possibilities to improve human life, or also, in some respects, to deteriorate the conditions of the human species. Biotechnologies developed in the 20th century hold possibilities to improve human life or deteriorate the conditions of the human species. The discovery of the structure of DNA in 1953 spurred a revolutionary progress in biotechnology. Biotechnology holds prospects for the welfare of humankind, but also portends detrimental outcomes. Among their benefits, identification of the genetic base of disease, eradication of genetic diseases, development of therapies, prolongation of human life, and much more. Few among their unfavorable implications are human cloning, eugenics, and biohazards. Once again, eugenics is the science of improving human population by controlled breeding to increase the occurrence of desirable, heritable characteristics. Among biotechnologies developed in the 20th century are several to be noted here. They include recombinant DNA, gene editing, stem cell research, and reproductive cloning. Some of these biotechnologies hold prospect for the well-being of the human race, but can also have detrimental outcomes. Among their significant benefits are identification of the genetic base of disease, eradication of certain deadly diseases, development of drugs and therapies, prolongation of human life, refinement of human genome, and much more. At this point, I wish to offer only a few words on the intricate and elegant recombinant DNA biotechnology. It has been a tremendous boon to humankind. Thus, it warrants brief explanation. This brilliant, intricate, and significant recombinant DNA biotechnology has made possible effective treatment for a number of diseases. Recombinant DNA biotechnology. Recombinant DNA technology is an elegant biotechnology which has been a tremendous boon to humankind. Thus, it warrants brief explanation. This brilliant, intricate, and significant technology has made possible effective treatment of a number of diseases. It has been a boon to humankind. Briefly, two sets, that is different segments of DNA, one the host DNA and the other the desired DNA, are combined under specific conditions. This diagram shows the recombined structure. The diagram shows the two segments of DNA in two colors. This is known as a chimera because it is the result of two genetically distinct sources. Recombinant DNAs are treated such as to significantly increase that number. By this means, through this technology, a large amount of the desired product coded by the inserted DNA can be obtained in pure form. This technology has been life-saving. Through this technology, vital therapeutic agents can be obtained in large amounts in pure form, such as blood clotting factor 8, deficient in hemophilia, insulin, and an ever-growing list of therapeutic agents. The importance of significant biotechnology cannot be overemphasized. This fascinating technology is revolutionary. It provides vital agents for the treatment of a number of diseases. Thus, we can conclude that recombinant DNA technology is beneficial, therapeutic, and highly moral. The early recombinant DNA experiments. At this point, I wish to introduce the humor, the significance, and the ethical concern which have been historically associated with this type of experiment. The early recombinant DNA experiments were historic and significant in several ways. In 1972, Paul Berg created the first recombinant DNA chimera at Stanford. It was the result of combining DNA from two genetically distinct sources. 2 years later, Cohen and Boyer devised an amusing chimera. They transferred frog genes into a bacterial cell. It became known as the Frog Prince experiment. There was fascination and humor therein, because when Cohen was asked how he had identified the bacterium expressing the frog gene, he jokingly said that he had kissed the bacteria to see which one would transform into a prince. Thus, this significant experiment has become known as the Frog Prince experiment. It was Frog Prince as it relates to a story in a well-known fairy tale by Grimm, whereby through a kiss, the frog is magically transformed into a handsome prince. Aside from the strange humor therein, learning has been gained from this experiment. Furthermore, this type of experiment raised alerts about the ethical issue. Much learning has been gained. Importantly, from recombinant DNA experiment, much has been gained toward the treatment of disease and human well-being. Aside from the seeming absurdity of mixing the DNA from two species and the humor therein, the development of this type of technology, that is recombinant DNA technology, has been a boon. This technology serves for the synthesis and production of a number of vital therapeutic agents in highly purified form. Indeed, we can build a strong case, once again, that such technologies are a boon to humankind. Such technologies serve for the betterment of humankind. Thus, they can be highly moral and ethical. Thus, through recombinant DNA research, much has been achieved. As an example, in order to obtain insulin, essential in diabetes, one route was to obtain pancreas for a large number of cows. Pancreas is where insulin is produced. It was necessary to grind the pancreas, isolate and purify the insulin fraction. However, employing recombinant DNA technology, large amounts of insulin can be obtained in highly pure form. We can nevertheless rightly ask, can such technologies lead to unethical outcomes? This first experiment of DNA raised alerts about the ethical issues entailed in such experiments. It is heartening that the scientists themselves were at the forefront. Paul Berg, a leading researcher in the field of recombinant DNA and one of the recipients of the 1980 Nobel Prize in Chemistry, first expressed concern. Paul Berg, the creator of the first DNA chimera, was alarmed by the inherent hazards of recombinant DNA technology. Together with several other noted scientists in the field, he raised serious concerns. This led to 2 conferences in Asilomar, California. To the second conference in 1975, lawyers and journalists were also invited. There was transparency. Berg summarized the data and outlined the scope of the problem. It was a volatile meeting. Lawyers spoke, scientists spoke. A unanimous decision was reached to place a temporary moratorium on recombinant DNA research. Other powerful genetic technologies were developed in the 21st century. These technologies have spurred scientific and medical revolution. One was the decoding of the entire human genome in 2003, at the beginning of the 21st century, a bare 50 years since the discovery of the structure of the DNA molecule. This was the Human Genome Project, an international scientific research endeavor. It was formally launched in 1990, cost $3 billion, and was declared complete in 2003. We now know that the human genome includes over 3 billion letters of DNA. It consists of 23 pairs of chromosomes and codes for 20,687 genes in total. It accommodates enough variation to make each of us distinct, and yet enough consistency to make each member of our species different from other species. The decoding of the entire human genome has been significant and extremely beneficial to human well-being. Once the human genome was deciphered, several significant technologies developed, notably among them gene copying. Several significant fundamental technologies finally led to development of CRISPR, an elegant technology for gene editing. This work is revolutionary. There has been equal case on who should receive the patent for it. This powerful technology enables genomic surgery, genomic editing. CRISPR-Cas9 is well known to the public. The name is familiar to the public. It's a technique for gene editing. The possibilities of CRISPR are immense. If we know the DNA sequence related to a gene, related to a given organism, we can perform genomic surgery. The development of this technology for gene editing was the outcome of the research of a number of scientists, among them two remarkable women scientists at the forefront. They were the ones who made the most significant contributions. The 2020 Nobel Prize in Chemistry was awarded to two women scientists for their groundbreaking work on gene editing. That is, Jennifer Doudna from University of Southern California in Berkeley, and Dr. Emmanuel Charpentier, the director of Max Planck Institute. There were also other researchers who made significant contributions to this field. The possibilities of CRISPR are enormous. Among these possibilities, some offer remarkable benefits with significant implications in the treatment of diseases and abnormalities. Some have worrisome implications as to their misuse. This technology enables genomic surgery. With genomic surgery, it's possible to chop, delete, and add to and revise the gene at will. This technology has the potential to eliminate certain genetic-based diseases. It enables development of novel therapies. However, this technology also has worrisome implications. Among them are eugenics, already mentioned, and creation of biohazards and their release into the environment. On one hand, these biotechnologies cannot be overlooked nor eliminated, as they can offer numerous benefits to humankind. Yet, on the other hand, ethical and spiritual concerns continue to engage biologists and ethicists and others. Significant ethical and spiritual concerns continue to face us with ethical and spiritual implications. Among these concerns are eugenics, already discussed, gene enhancement, which enables the selective enhancement of a special trait or characteristic. Thus, it can result in inequalities, evolution of a super race and underclass, as predicted by Aldous Huxley in his dystopian book, The Brave New World. Some associate this technology with creating what has been called designer babies. And then among the ethical and spiritual implications included is also reproductive cloning. Reproductive cloning uses two possible approaches, either use of somatic cells, which is any cell other than the germline, or the use of embryonic stem cells. Both of these approaches to reproductive cloning provide benefits but also have ethical implications. In view of the lure and the fascination of the most recent powerful genetic technologies developed in the 21st century, we can rightly ask whether scientists can or will continue to self-regulate. The ethical and spiritual implications of these technologies compel this question. Can scientists self-regulate? And to what extent should spirituality, moral principles, and ethics come into play? How can they be enforced? How can they be regulated? Several fundamental ethical concerns emerge. Indeed, at this point in time, we are at the crossroad of science, ethics, and spirituality. This compels us to reflect on the standpoint of Baha'i Faith in respect to the harmony of science and religion. The standpoint of Baha'i Faith. This compels us to ask the question, are we moving closer to the brave new world of Aldous Huxley? Mentioned earlier, I made reference to this dystopian book of Aldous Huxley, The Brave New World, wherein Huxley anticipated ominous circumstances and scenarios. Can these indeed become realities? Or we can ask, are we advancing toward the vision foreshadowed by Shoghi Effendi, the appointed guardian of Baha'i Faith? We have the guidance through the writings of Shoghi Effendi in the World Order of Baha'u'llah regarding a world system guided and inspired by the vision expressed through the following guiding statements in the World Order of Baha'u'llah. That is, the enormous energy dissipated and wasted on war, whether economic or political, will be consecrated to such ends as will extend to the range of inventions and technical developments, to the increase of the productivity of mankind, to the extermination of disease, to the extension of scientific research, to the arising of the standard of physical health, to the sharpening of the refinement of the human brain, to the exploitation of the unused and unsuspected resources of the planet, to the prolongation of human life to the furtherance of any other agency that can stimulate the intellectual, the moral, and spiritual life of the entire human race. Fundamentally, such reflections lead to the reality of our time that there is significant imbalance, discordance between advancement in science and advancement in spirituality. Spirituality has not advanced in line with advancement in science and technology. These have not been in synchrony. Science and spirituality have not advanced, progressed in tandem. We have progressed materially, but not spiritually. Some may even hold that we have advanced in science and technology, but have indeed regressed in spirituality. Material and spiritual advancements go hand in hand. Both are essential to the progress of civilization. Material civilization is like the lamp, but the spiritual civilization is like the light in the lamp. This lamp without the light is a useless thing. Now, at the conclusion of this presentation, reflections on the Baha'i principle of the harmony of science and religion, the ethical challenge posed by biotechnology, I wish to state that this presentation was centered on reflections on the fundamental Baha'i principles of the harmony of science and religion, and the ethical challenge posed by the principle of biotechnology, notably by the biotechnology of genetic engineering. This presentation was based on some of the current knowledge in the field of biotechnology. With the passage of time, increasing development in novel biotechnologies are bound to provide yet greater benefits. They're also bound to pose yet new challenges. Once again, the viewer will be left with that fundamental and enigmatic question. Does biotechnology serve as a boon or gloom for humankind? Boon? On one hand, novel biotechnologies, through the great benefits they impart, are bound to serve as boon for humankind. Yet, on the other hand, they may pose more serious concerns as a gloom for humankind. Biotechnologies hold enormous value, yet they can also pose serious challenges. May humankind advance in line with that higher level of spirituality, conducive to an ever-continuing boon toward human well-being and felicity. At this, the conclusion of this presentation, I wish to express my appreciation to the viewers of this presentation for their interest and investment of their time in listening to and viewing this presentation. I appeal to the viewers to reflect on the implications of the fundamental Baha'i principle of the harmony of science and religion for this and future generations. Again, as biotechnologies continue to advance, new questions, new challenges will continue to engage and intrigue humanity. Will such more advanced biotechnologies serve as a greater boon or a greater gloom to humankind? Both of these will continue to intrigue us, but also to serve humanity.